So first things first, we'll go to an introduction and a disclaimer, and then we'll discuss some PG pathways and do some presentations, so speakers will present. We'll have a short Q&A session, so if you have any burning questions, you can ask them. And then we've got some pizzas and drinks and stuff out the back, and we can do a bit of networking at the end. So just a disclaimer that the views shared in this presentation basically don't reflect the views of the University of Auckland or AUBSA. And if you want official advice about grades and stuff like that, see the Science Student Centre or the Biology Student Centre. So we've got a few speakers tonight. We've got me starting off in advanced ecology, then we've got Wednesday, and Christine, who are doing marine ecology. Then we've got Yinko, who's doing space biology, and we will finish off with James, who will talk about molecular biology. So if you really want full academic help, here are some places that you can go to. Academic advisors at the Science Student Centre. There's also undergraduate advisors in your school and department. So you can go see the bio department, and they can just basically help you with all these things. So course planning, making a degree, and choosing your major and stuff like that. So basically, there's a few postgrad pathways that you can take. So most of you would be doing a BSc. So basically, after you finish that, you can either go for an honours degree, which is 1 year, or a PG Dip, which is 1 year. So honours is sort of a mix of courses, and then you have to write a dissertation. And in a PG Dip, you just have courses. And then basically from that, you can go on to a master's, which is a full research project. Or if you do well in your honours, you can progress straight to a PhD. And there's the new program, Bachelor of Advanced Science, which is a 4-year program. And from that, you can either go on to PG Dip or Masters, or if you do really well, you can progress straight to a PhD. So a bit about me, I'll start off. So I basically graduated from Auckland Uni in 2017 with a BSc in Ecology. And then I took 6 months off to travel, and then I started a PG Dip at Auckland Uni in biosecurity and conservation, which is sort of quite a natural pathway. And I focus on all the plant ecology papers. And then I also, in the middle of my PG Dip, I completed a summer studentship in 2018, 2019, which basically focused on cold-y drought responses. And then I started my master's, which was like focusing on what sets the southern limits of different plant species in 2019, and finished last year. I did a bit of work after that, and then I'm starting a PhD in May 2021, which is focusing on basically the survival of cold-y in the face of climate change and dieback. And I've got a few tips for undergrads myself. So postgraduate diplomas are very similar to doing an undergrad, but they're basically a lot better because you interact with your classmates a lot more. You have more field trips and lots of like group work and stuff, which is actually really fun. And you actually make friends with quite a few people in your course. And but it is quite a bit more work. Like the assignment, you don't really have exams, but the assignments are a lot bigger, and they tend to be bunched up at the end of semester, so it can get a bit tricky. A semester off is actually a really good idea because it allows you to sort of like get your head around what you want to do in postgrad. And especially if you're not certain on what area you want to get into. Summer studentships are really, really good for finding potential projects. They basically get you in touch with supervisors, and they get you in touch with supervisors and the wider research community. And they just you also work quite a bit with masters and PhD students. So you sort of get a feel for what that's like before you even start, which is a really good bonus. And also, you don't need to find like an exact master's project when you basically go into a PG Dip. But sort of have a little think about what area you want to get into, like whether you want to get into plants or animals or molecular biology. And then in your first sort of half, your first semester, you want to have a little look around for supervisors and stuff and just figure out what your project's going to be. And usually in your second semester, you take a paper called 761, which is like a master's proposal prep paper. So that's when you do a little presentation on your master's, what you want to do and stuff. And yeah, and you do like start to suss out your project, which is quite nice. But yeah, our next speaker is Wednesday Davis.
I have a question for you first.
Oh, yes.
Your Cody Dieback stuff, are you doing cellular level stuff or more microbiology work? Can you tell us a bit about your master? Oh, sorry, your PhD.
So I'm basically looking at a big scale type thing. So it's basically looking at the trees themselves, like on a whole stand level. And we're using like growth rates, mortality rates to build like a baseline model of coalty growth. Like we're just seeing how they work in a normal setting without those stresses. And then we're going to add dieback, not add dieback, but sort of simulate dieback in the models just to see if they'll survive. And then what that hopefully should do is if there's some variation between different areas, you can sort of identify coalty forest stands that are more susceptible or less susceptible. It's quite exciting. But yeah, there'll be a lot of seedling measurements and plot work and stuff. But yeah, any other questions? Oh, sweet as soil. Yeah.
Sorry, guys. So just while we're looking at that, hi, my name's Wednesday and I'm a master's student here at Auckland University. Oh, yeah. Oh, that's the one I look at.
Oh, okay, so
so my presentation is a little bit longer than Toby's, since I won't bore you too much. I just have it off.
Oh, good.
I'm technically challenged, apparently. But yeah, I'll get started anyway. So I started here at Auckland Uni in 2016. I'm from Tauranga and went to Mount Maunganui College. So as you can probably tell, I am one of the marine people. So I grew up right by the beach, spent all my summers right next to Mount Maunganui Beach, very beautiful, lots of fish, lots of marine life. And what was really cool about growing up in Tauranga is that I really got to experience a lot of the issues that were also there, but I'll tell you about that in a sec. Confusing me. Awesome. Yeah, so I'm a master's student at Auckland Uni. So I've got a pretty cool project. It's kind of the dream marine biology project. I am working on whales, dolphins, and seabirds. So I get to go out on the boat. The university has its own boat, which is really cool. And I get to go out into the Auckland harbour, scoot around on the boat and look for workups. And what I do when I find them is I fly a drone above them, capturing the view from the sky, kind of like a bird's eye view. And then when I get back to the lab, I actually am training AI, so artificial intelligence, which is pretty cool. And hopefully that will speed up the process. I did a postgraduate diploma here at Auckland, as well as a Bachelor of Science. So a little bit about my background. As I mentioned before, grew up in Mount Maunganui, fell in love with the ocean, but I also lived in China for 3 years. If you've seen pictures of China, you probably know it's really, really polluted, not just on land, but also in their rivers and oceans. And I love exploring, learning, and discovering how things work. So pretty much all of that led me to do science. Here is a picture of Qingdao, which was the nearest beach to where I lived. Pretty nasty. That is a whole load of algae, which grows when there's too many nutrients in the water. And here is a really awkward photo of me when I was about 13 years old. Very cool, I know. And that is us cleaning up oil from the Reena disaster. So you may remember, big boat crashed off Tauranga and all of its oil went onto the beaches. So even when I was really young, I saw seabirds dying from getting coated in oil, as well as lots of fish and jellyfish and whatnot. I also did lots of sciencey things at high school. I went to cool leadership camps and science camps, including an environmental leaders one, Hill Laboratories, Waikato Uni, and Powering Potential. So that's kind of how I got into science. I also did a lot of scuba diving, which is a great way to kind of learn about the environment by being in the environment. So this is my Nana and Papa wearing some very awesome retro scuba gear. Doesn't look like that anymore, but safer. And this is me working at Kelly Tarleton's as an aquarist. So basically in high school, fell in love with the marine environment. When picking a career, if you love the ocean, you love learning about marine critters, you love learning about how the ocean works, why not do a Bachelor of Biomedical Science? If you're smart and you like helping others, you want to have a higher purpose, why not become a doctor? I did not become a doctor. I really didn't like the content, wasn't getting good grades, really didn't want to work in the healthcare sector. So I changed to marine biology. So these are just a few photos from my undergrad, learning about behavioral science, ecological physiology, fish taxonomy, and also being involved in some clubs. So yeah, that kind of message is it's never too late to change. If I had done biomedical science, I probably wouldn't have got a job as a biomedical scientist. So after switching to marine, I got to work some really cool jobs in the industry, including being an aquarist, so aquatic zookeeper, working at Auckland Council as a research monitoring and surveillance student, and working at Rent-A-Kill, the pest control company, on the National Invasive Ant Surveillance Program. So if you do something you love, the jobs are going to be there. If you're really passionate, you work really hard, you can find really cool opportunities, and you never know, if you try something different, you might enjoy it. Turns out being good at fish taxonomy makes you pretty good at learning ants. Sorry. There we go. So some of you are probably getting to think about your papers. I recommend having a look at some now so that you have the prerequisites before you get to third year and fourth year. So what I did was lots of marine papers, but also some biological sciences as well, with a little bit of fun environmental science modeling in there too. So the biggest difference between postgrad and undergrad is the way the papers are taught. Instead of having a few hundred people in your class, like undergrad, it's usually about 20. So really small classes, a lot more critical thinking and developing ideas, not just memorizing fun facts. So when you're in PGDipSci, you'll also have to pick a supervisor. This is not my supervisor, it is a sea lion, but it's really good to have to think about now what kind of stuff you're interested in. So what qualities do you want? Do you want them to be a woman in science, someone who is LGBTQ, or maybe even a specific ethnicity? They're not just guiding you in your masters, but also they're a mentor. What do you want to get out of your masters? Do you just want a piece of paper, or do you want a bit of an experience? Don't be afraid to talk to lecturers. They're not scary. They get paid to talk to you. And talk to them early, not right before you enroll in your thesis. So my supervisors are Professor Rochelle Constantine. Why did I pick her? Awesome woman in STEM. She's done lots of really cool stuff. She works with big animals, not just mud and, mussels. And also she had a really cool project on the go. So I knew that she probably needed some help, and there was a little bit of money to do fun stuff. I also have Gaia Delaricia, who's also a cool woman in science. We worked together at Auckland Council, so she knew I was pretty cool. But also she's a really great seabird scientist. So I knew that she'd be pretty helpful if I did my master's on seabirds. So as I mentioned earlier, I work on cetaceans, seabirds, and praise, and use drones. So this is a brood as well. They're pretty cool, and I get to do my research with them. It's not loading. There we go. This is what my drone footage looks like. So I go out in the ocean, fly drones over top of workouts, and have a look at what they're doing. So these are New Zealand common dolphins, and there's a gannet flying just before. So what I'm doing is I'm taking snapshots, about 24 per second, and training the computer to look for these different critters. What does that look like? Sorry, it's gone a bit too far. This is what it looks like. So basically, I need about 5,000 of every single behavior for every single animal to train the computer what it is. So instead of me doing it myself, hopefully the computer will do it for me. So these are all common dolphins. You can see they're pretty pixelated, but the computer can work out what they are. But sometimes when it gets a bit busy, it's a little bit harder to see. Sorry about the audio. I was hoping it wouldn't be included. But you can see there's a lot more going on. A lot more animals, a lot more behaviors. You can see the gannets are flying, they're swimming down, they're sitting, they've got these little trails of bubbles. So when it comes to monitoring their behaviors, it looks a bit different. We've got flyers, we've got sitters, we've got diving under the water. So as you can see, it's really handy to have that AI. I've also seen some cool critters like breeders' whales, hammerhead sharks, and little blue penguins. But even though that's really cool, you have to get something out of your masters. So I'm trying to look at who's in the workouts, what are they doing, why are they doing it? From my masters itself, I hope to get some really cool ID skills, behavioral ecology skills, learn how to use AI in drones, and learn some statistics as well. So hopefully I'll be able to get a career in whales and dolphins. So you want to do postgrad? A little bit of advice from me. One is get a great support network. Neil is my friend. So friends are a great way to bounce ideas off people. It's nice to have good times and bad times, have a quick lunch, or even around for a yarn. So these are my friends I made my third year behavioral science class. I'm still hanging out. We still like each other, which is pretty good. Be open to new things. You never know where it might take you. All experience is good experience, and it's not too late to change. If you don't like your major, that's okay, you can change. If you want to add an extra degree or diploma in there, you can do that as well. Certifications. Certifications outside of uni, like first aid, scuba diving, VHF radio, maybe even foil driving, makes you stand out from the crowd, especially if you want to do animal science. End goals, have a think about where you want to be, chat to your professors and lecturers, make sure you're doing the right prereqs. I had to do an extra semester because I messed up my degree diploma. That's okay. And figure out your learning style early. Are you a mind map person, a rote learner, a listener? It's good to get it down now before you get into later on in your degree when it gets a bit trickier. Yeah, thank you for your time. If you have any questions, fire away. Otherwise, I do have my contact details. You're welcome to reach out. And yeah, thank you. Anyone have any questions before we carry on?
Oh, sweet. Thank you, Wednesday. That was really good. So up next, we have Christine. We'll be also doing marine science.
I don't have drone footage, so I'm not going to be nearly as interesting. Sorry. I was excited. Kia ora, I'm Christine. I started uni in 2015, so I'm feeling a bit old hanging out here now. I did 3-year undergrad, a bunch of details.
and then did honours, and now I work at Auckland Council. So I kind of pieced out of studying as soon as I could. Okay, so yeah, like I said, just like Wednesday, I was at high school and I was good at science and I wanted to help people. So I was nudged into doing biomed, and again, just like Wednesday, didn't love it. So I decided at the end of my first year of biomed to switch into biology and do something that I might get some better grades at and something that I would actually enjoy. Still kind of wanted to do pharmacy in the end and kind of kept changing my mind as I went until like halfway through my third year where I was like, yeah, that's not going to happen either. So stick with what you know, do biology, and sort of winged my way into a last minute honours project. So if you get anything from me, it's be prepared, don't make decisions at the last minute, because I just kind of fluked it and I'm really lucky that it worked out. So I finished my honours at the end of 2018. I ended up working on a project that is technically marine ecology, but I was under biology, more complications we won't go into it. But I was working with NIWA on one of their snapper projects. So I was focusing on juvenile snapper and how they grow in different environmental conditions. So essentially taking the little bone out of the ear of the snapper, grinding it down, getting its age rings like you would have in a tree, and figuring out how fast it grew based on where it was living and whether or not certain environmental conditions meant that it had a better chance of growing and succeeding in life. Like I said, I just kind of wanted to get done with uni, so I just did the bare minimum to get me a good standing for a job. If that's what you want to do, great. If you want to go on and do awesome masters and PhD projects, that's what you have to do too. But again, I managed to slip my way into an Auckland Council grad program, doing in their Environmental Services Department, doing science communication, and then kind of turned into my current role, which is a community conservation advisor. So if you look at where I started at uni and where I am now, they're different realms of reality entirely. So my point on that would be to be prepared for your plans to change and to end up in a career that you might not have thought you'd love, based on where you started. How do I do next? Oh, just the right. Oh, yeah, and that's me at the beginning of my study. They were just put it in sediment. Oh, sweet. Awesome. So yeah, pretty much everything I've already covered. Like I said, start thinking about postgrad early. Obviously Wednesday and Toby said that too. Yeah, give yourself the best chance of talking around to lots of different advisors and supervisors, and keep your options open. I ended up kind of cornering myself into one thing because I was like, I need to find a project now, I need to do this now. And luckily I ended up enjoying the project, but I could very well have ended up doing something I didn't love just because I left myself to the last minute. Think carefully about who your supervisors will be and how their sort of way of working might align with how you like to work. For myself, I like to know how everything's going to plan out, which is ironic given that I've winged it most of the time. And I would have liked to have a supervisor who was like super like involved and wanted to talk to me all the time and checking how I was going. I ended up with two awesome supervisors who didn't quite have that work plan, so it ended up being a little bit like I was just kind of doing it on my own, which might work for you if you want your supervisor to leave you alone. Find a supervisor who will leave you alone. If you want to find a supervisor who will work with you at every step of the way, have a chat to them about that before you start and make sure you set up how you want your working relationship to look. Like I said, be open to change, be open to change your career path at any point in your university journey and afterwards. Like I said, I'm not doing anything like what I thought I would be doing. So it's good to have goals and get excited about what you're doing, but also keep your mind open to other opportunities that might lead you to something that you really love. Not to be a pill buzz, only take bio, say 7th grade if you really have to. It's stats paper, it screwed my GPA, I didn't even need to do it. So just check with your supervisor, they will tell you if you actually need to do it, and only do it if you really have to, or if you actually really love stats. Just, it was not a good time. And don't forget to have a life while you're at uni. I'm sure everyone who's not at uni will tell you that, but I've lived in Auckland for like 6 years and have only just actually started going places and doing things. And so people can all be like, what should we do? And I'm like, I don't know. I spent my whole first 4 years here. So make sure you have fun. And like Lindsay said, have lots of good friends around you and enjoy it while you can. Yeah, that's me. Questions? Oh, two, okay. Yeah.
So just more on your current role at council.
Yeah.
Do you mind telling us exactly what your role entails?
Sure. So how far back do I go? Okay, so as part of Auckland Council, they have a new project going on called NETA, which is Natural Environment Tailored Rate. Bunch of extra money coming in just for environmental projects, which is really awesome. And part of that project is an initiative to get regular Aucklanders who might not already be involved in conservation work or might be sort of like thinking about it and sort of take them and get them more involved. So we work with like volunteer groups that are already up and running and want to get more public, like normal Aucklanders involved in what they're doing. We give money to people to do training and grants to help run their projects. So yeah, I run a grant program with a bunch of money to give away. I run training programs for volunteers, so first aid training, tracking training, health and safety training, all that kind of fun stuff. I do comms, run Facebook pages, write newsletters, contract management, boring stuff. Yeah. So essentially just trying to get more Aucklanders involved in conservation in their own space. So trapping in their backyards, identifying and pulling out pest plants, and planting native trees. Yeah. Did that? Yeah. Did you have a question? Yeah. Yeah, so it's called advanced biological something, but essentially it's stats and it's coding with R. And ironically, when we did a test or like an assignment, and our teacher, whatever they call lecturer, said, "What did you learn the most from this assignment?" We're like, "We learned how to code in R," and she was like, "That's not the point of the assignment. We were supposed to learn stats." I was like, "Yes, but you didn't tell us how to code it then, so we had to learn how to code to do stats." It was, yeah, she was cool, but it was weird. Yeah. Awesome. I'll be around if you have any other questions. Sweet. It's only say just need to sign it over a little bit more.
Okay, sweet. Sorry about this, guys. Sweet, you got it. Sweet. Yeah, just
You guys can
talk for.
Yeah.
I think almost everyone that photo stayed on the yeah, she's teaching still amazing to see.
I just like saw him peeking in your photo and I miss it.
He's working on stuff. Yeah.
Did he like drop off the places that
He kind of did.
Yeah. Yeah. Me and Elise.
At least it's hot. Taking very nice. Yeah. Amazing.
I was messaging her before.
Yeah.
Like being on campus, like
that's the big place. But do you know someone in Cape Jesse?
yes.
Yes.
Yeah.
From high school. Yeah. So before she was in the department of sustainability, she was someone that like sustainability leadership stuff regionally.
Yeah.
So I went to a bully for that.
Right.
And then basically started running bullies alongside her. And I still keep in touch with her. Yeah.
Basically, it's something.
I saw that you were talking about
So if you want to go right, you just if you want to go right, just tap on that. Yeah, thank you.
Hello, hello. Hey, everyone. So my name is Yan Kai. I'm currently a master's student here at the School of Biological Science, also at the Space Institute, the I don't know how to say it, but yeah, this today will be mostly about my master's experience. And so really, so the title I'm allowed to give you is that I'm looking at the effects of simulated microgravity on a particular bacteria. And so this is actually a self-proposed project because there's no expertise. There are no supervisors in this area. So something I had to do was actually write down the experiments and then started going to supervisors saying, put money onto my project, please. And after around 5 rejections, I was able to find a supervisor that supported this project. And so that's pretty good. Otherwise, so currently I'm going to be going through an embargo on the project, meaning the thesis will not be public. And so I can't actually talk about it. So what I'm going to talk about is the other things, all the other cool stuff that I did during my master's. And so I sort of start off by talking about something I'm a part of, so the Space Generation Advisory Council. It's a non-governmental organization that supports the United Nations, but it's really tailored towards university students and young professionals interested in the space industry. So yes, I'm the national point of contact. And last year, I don't think many people heard about it, but I organized the webinar, Creating Opportunities Through Diversity in the New Zealand Space Industry. And so we actually featured an old woman webinar. So we got Barbit from the Extraterrestrial Power, Hazel Deacon from Sarah, which is a Earth observation company. I've got Priyanka. She was named the top 50 woman in engineering, but she was also the top 72 candidates for the Canadian Space Agency to become an astronaut. And the Mahima, who's in Auckland, she works at Rocket Lab. Another thing I'm a part of through the SGAC, the Space Generation Advisory Council, is that I'm a committee member of the Space Medicine and Life Science Project Group. So actually I ran a series of webinars last year as well. One of the things was an introduction to the area of space medicine and life science. So we actually featured two plant space biologists. And so one of them talked about immune system, plant immune system in space flight, as well as water retention, as well as alternative splicing, transcriptomics, alternative splicing. And then so Alba Serrano actually worked on space neuroscience. And then we also had Grand Systems who organized payloads into the International Space Station. And he talked about space development in Asia. Another webinar I worked on was a shift towards the European researchers. And so we had Trisha, really wonderful person. She worked on tumors in space. And then we also had Stefania, that she looked at spatial transcriptomics, as well as William, who worked on mitochondrial dysfunction in space flight. Another thing that I'm currently working on, but there's currently no dates for it, is that we're going to have a webinar that's going to feature a Japanese female astronaut, as well as a Taiwanese food regulator, and then also a space nutrition company from Thailand. And another thing I was involved in was in the Taiwan's National Space Program, the ICAS 2020 competition. I coached the high school students on designing experiments for the International Space Station. Another thing was, so the European Space Agency had a business incubator for space startups. And so I provided guidance to an all-female startup on space pharmacy. So I won't talk too much on that. Another thing I did was I personally gave a lecture on an introduction to space medicine and life science. And this was for Sri Lankan students. They had specifically requested someone to teach them this area. And another thing that was a bit more recent was I gave some advice to a Singaporean startup that looked at vacuum-based biological experiments. It's a bit interesting because the startup, the Singaporean that made the startup, he tried to send himself up to space and make himself the first Singaporean in space. That didn't actually work, but he's going to try again. And so maybe we'll hear about it in a few years. And something I did also last year was the Asia Pacific Space Generation Online Workshop. So I was the moderator for the Space Medicine Workshop. So we would basically talk about using space medicine to address COVID-19. And so some of the things we talk about is printable medical device, telemedicine, but also health and wellness, which is essentially space psychology, if that's an area that you're interested in. There was a workshop organized by MIT. This was called the Human-Computer Interaction Space Exploration. And so basically, my team and I, we proposed of using wearable technology with soft robotics to help mitigate the effects of muscle degradation in lunar colonization. So a good thing is our paper got accepted. There was around 50 submissions and 20 teams got submitted. And it's a good thing we were one of the people that got submitted, that got accepted. Another thing was the International Astronautical Federation, which had this year they had the GLEX 2021 conference, which is in Petersburg, Russia. And so we submitted a a submission on AI healthcare for diagnostics in space. It's a bit interesting. So we got in, but right now Russia's still getting 20,000 cases per week. So we actually don't know who's actually gonna, who from team is actually gonna go to Russia for obvious reasons. So we might not actually be able to present essays. And I guess my lesson is really, just reach out to people. There's a lot of, even in Oakland, there's a lot of expertise in the space sector if you're interested in that. And I didn't talk too much about my master's, but something I did notice is a lot of people do get stressed from their master's. And so really helping other people is probably one of my main messages, because when you help other people will be more inclined to help you. And so that really helps out with your year-long or project. Also, if it's a PhD, it's a 3-4 year project. And I'm not really sure this is a good advice to give. But when I say there's always time for more, this really depends on the type of person you are. Some people won't be able to handle much more, but you can always still succeed well in life by just focusing on your research. And yeah, definitely start writing as soon as possible, because I'm getting the bad consequences of not writing as soon as possible. I was like, that's all really. Yeah, thank you for listening.
Hi.
Yeah, go for it.
Oh,
yeah.
Sorry. So you really touched on how you got to where you got. Do you mind touching on how you reached this area that you're currently in? Like, what was your journey from undergrad to postgrad?
Oh, right. Yeah. I only noticed after I was watching their presentation. I was like, wow, I didn't talk about my background. I grew up in Auckland as well. So going under, so I went through the New Zealand education. So I guess I was, when I was a bit in high school, but also young, I was interested in astronomy. But I think I frequently visited the Star Dome as well. But when I was looking at what they actually did to look at the Star Dome and what they did when they were looking at planets, while the planets looked cool, I wasn't really sure whether that was something I wanted to do. And I actually gradually forgot about it. And when I started uni, I started with a music degree. I wanted to compose music, but that didn't happen. So I switched to science, changed majors a couple of times as well. And it ended up just finishing a BSc in biology. I went to work a little bit, but then I didn't enjoy work. So I came back to do postgrad. And my postgraduate diploma, and as some of these you have already talked about, you have to do a master's proposal. And at that time, I still wasn't too sure what I wanted to do. So, but it was also at that time I remember that I was interested in space. And then for my assignments, I was also writing about, because I didn't like the assignments, I told the lecturers, could I write about space biology instead? And they let me write it. And I think it was at that point I realized, hold on, we could do this here in New Zealand. We don't need to go overseas to do these research. And I guess at that point, it was kind of like, just think about, okay, it was, when you're looking for your supervisor, like I might as well go all or nothing. I just wrote the proposal and went around, start asking for supervisors and see who wants to do this. Yeah. I hope that's a good answer to that.
Thank you. Sorry, and who is your current supervisor? Or I do not want to say that.
Oh, no, I can't say that. My supervisor is AUBSA. Yeah, as I already pointed out, he doesn't have the expertise in this area, but you don't necessarily need the expertise in this area, because no matter what, these expertise has to start somewhere. Obviously, it's a lot easier if you have a supervisor that already knows this, but I saw it works out for me, because rather than having been taught an area, I was able to kind of, I guess, figure my way through and come up with my own idea of what this field is about. Yeah.
Thank you.
Any other questions?
Those webinars are super interesting. Are they like open to the public?
Oh yeah, yeah. We just had a huge trouble trying to find people to get interested.
Yeah.
Yeah, like particularly the one about the diversity in space, in the space sector. We struggled considerably on that. Yeah. Anything else?
Oh, sweet. Thank you for that. So last up we have James.
Cool.
That's probably the easiest. Okay. Right, what I'm going to do. I find your place to in this room. So it should theoretically just work. Ta-da! Yes. Okay. So I'm going to try and not stand behind a thing because that's been my life telling people not to. So wow. That's right. This is a university issued computer. It doesn't work. Cool. So hello. I'm James Hucklesby. So I'm a PhD candidate at School of Biological Sciences. So I'm just in that phase where you kind of realize that PhDs take a really long time and that the money only lasts 3 years and that in 6 months I'm not going to get paid anymore. So I'm kind of in that phase. So I'm kind of right on the other end before you have to hand in this massive thesis and everyone calls you doctor and you have to go around pretending you know what you're talking about. So that's roughly where I am. So I started off, I did my undergrad. I started off in medicinal chemistry because I thought that would be a really cool thing to do. Turns out I really don't like chemistry, which was a problem. So I did the opposite of everyone else and I switched into biomedical science. And then I've done a few summer studentships and bits and pieces along the way. And I'm still here and have been for a very long time now. So just for context, how many of you guys are third years? Almost all of you. How many of you know what you're doing next year? Excellent. We sort that out. So to start with, because I have research and I'm important, I'm going to tell you about it. So this is a subject everyone used to go, oh, who cares about the immune system? In the last 18 months, everyone suddenly cares about the immune system. So I'm going to tell you about it. So this is the inside of your blood vessel. And because I'm a cellular biologist, everything is colored boxes. So this whole presentation is full of colored boxes. So this is the blood vessel wall of the endothelium. So this is basically the cells that line the blood vessels all around your body. And there is blood flowing past it. And your blood is filled with a whole lot of cells. We call those T cells. And they're the ones responsible for immune responses. So let's say Neil has a nice cough and coronavirus gets released into the room. We're all going to start dying of coronavirus until your T cells talk to some other cells and start making antibodies. How many of you guys are actually biomedical scientists? Excellent. I can say whatever I want and no one's going to care. So here comes a T cell and you can see it just goes past. So you have somewhere in the region of 1 billion or 2 billion, if you're feeling a bit sniffly, T cells in your body, which raises the question, how the hell do these T cells know where to go? And the answer to that, one of the many answers to that, is a protein called CCL21. Now, when your cells are upset or when you're near a lymph node, the CCL21 gets presented on the surface of the blood vessel, and then if a T cell comes past, it can sense CCL21 and it will stop. And then after a while, it will have a clamber out through the cell and go and find some COVID or cancer or whatever else that's around that your body doesn't want. So how do we know this? Well, this is an experiment that I set up during my honours year, where in the top row here, we have those endothelial cells. these are a cell line. These are human cells that I've grown in a dish. And then on the bottom here, the top ones have CCL21, the bottom ones don't, and we've grown these. So just for scale context here, that's about 3 human hair widths across. And we image this with a very flash microscope up the road. And fortunately, it's relatively easy to get your hands on T cells because you can just bleed someone. So I've done that. And then so you can see them, I've actually dyed them green. So if I play this video, I know it's not a drone video, but it's the next best thing. You can see if it works nicely. No, I did test it and it worked beautifully. Here we go. You can see, so those are millions of T cells looking past, and you can see more are stopping at the top than at the bottom. And there's computer software that counts it and does stats and all that kind of fun stuff. So that was pretty much my honours year. I now do a whole lot of research around the brain, all that kind of stuff, but we haven't got an hour, so I'm not going to go into that. Instead, what I want to do, because I'm a bit further on than the others, is try and give you a bit of a bird's eye, broader view of what we're trying to get you to do when you're a postgrad student. So fortunately, I'm in a position now where I've been around the faculty for almost 4 years. So I've kind of had a little bit of insight into what supervisors look for, and I've seen some honours students suffer, and all that kind of thing. So what I'm trying to do here is just give you that really broad overview. So to start somewhere really simple, this circle represents the entirety of human knowledge. So rather a large topic. So this is everything that is known to all of humanity ever. And as I'm sure you can understand, that's a relatively large circle. So how do we go about teaching you this? Well, the first step is the central one. So the blue dot in the middle is what you learn kind of in primary school and kindergarten, which is things like talking to other people and using the pen, all those kind of really basic skills. Then you go to high school and we kind of give you a very light overview of most things, right? Most people have a working knowledge of some science and some English and some maths, right? So that's your really broad overview. Then you go to university and you get a circle like this. This is kind of what your undergrad degree looks like, right? You guys think your undergrad degrees are really specific? We purposely don't make them specific because we don't want a whole lot of experts on T-cell biology because there aren't that many jobs in T-cell biology. So what we do is we give you this really broad background with a little bit of expertise. So if you have a BSc, the idea is that you should have the skills to go out and get a BSc in anything else by yourself, right? So most of what we're trying to teach you as an undergrad is, yes, there's topic-specific stuff in that, but it's skills like learning how to write an essay and write a lab report and present to the public, all those kind of basic skills that we're trying to get at. Then we start to get into the postgrad realm, and unfortunately this projector is not great, but this one here kind of represents where we're trying to get you to in an honours degree or a master's or a PhD, right? You're starting to become a real expert in your area, right? You now know a lot about a specific area. So once you get into postgrad, we start giving you specific papers to look at. We start really narrowing down what you have to study to only what is really important. And we stop teaching you those more general skills because now you're a postgrad, you're a adult. And then this red bit represents a PhD. So the first kind of year of a PhD is great because you have all the time in the world to learn about a subject that you're really interested in. And so very quickly you get all the way to the edge of that circle. And the next bit's quite small, so we're going to blow up on that section. So this is like the end of the first year. Then the second year, you spend whacking your head against that wall and it does everything it possibly can to stop you. So cells will die, equipment will fail, stuff will just flat out not grow. You learn that in fact, things people say they can do in the literature, they can't actually do. And then you fly to the States because you think, oh, it must be me wrong. You find out they can't do it either, even though they said they could, right? So you spend this whole year whacking your head against a wall. And then eventually, towards the end of your PhD, this happens. There you go. That's a 250-page document right there called a PhD. So when you have a PhD,
you are very, very good at one tiny little thing. So if you want to ask me a specific question about how CCL21 interact with T cells on the surface of a bromine epithelium, I can answer your question. If you want to ask me about something slightly to the left of that, for instance, a T cell in the tonsil maybe, wouldn't have a clue. That's someone else's PhD, right? So when you start to get into postgrad and you start to get into PhDs, you get really, really deep, but really, really narrow. So the downside of that is that all the time you want to talk about T cells because your world looks like this, right? This is my world. This is the T cells in here. I get very excited about them, which is great because I know things that nobody else in the world knows. And I get to go to my supervisor and feel very smart as I explain to them that I was right, which is cool. The downside of this is that that's what I know, the scale, right? And the rest of the world looks at me like, why are you on about those T cells again? I'm like, because they're exciting. Everyone's like, no, we want to talk about, sailing or Easter or any of those things, right? So that effectively is where you're heading if you want to do a PhD. So that raises the question, should I do postgrad? Which I'm sure is sitting on the top of everybody's minds at the moment. So I'm going to try and give you a couple of pointers. If you want to do a postgrad degree, and that's really doing that move, really you want to find something that you are really excited about. Because postgrad becomes less about transferable skills. Yes, you still have some of that, right? PhDs go into all kinds of things that are not in their area, but it becomes less about that and more about your specific stuff. So firstly, you need to find something that you find really exciting, right? So be that T cells, be that dolphins or whatever. I have a mate over in SBS who has really strong views on a particular transcription factor in T cells. Really strong views. Like we had to separate him from someone over in OGH, which is the staff pub the other day, because they were having an argument, right? So if you have a topic like that, or you have a really deep interest in something, go do postgrad. Do that, right? That is what postgrad is for getting really interested and excited in something. Do not go into a postgrad degree if you want to be called doctor, right? You're not going to make it. So try and find something that you're really interested in. The second thing you should do is ask, who is doing that thing? So it looks a lot from the outside like the lecturers have their own little kind of research programs and they will run. It's very much not like that from the inside, right? So one thing you find once you're in a research lab is that good students are actually really hard to find, right? And you guys are, I'm going to guess you're all good students because you've turned up on a Tuesday evening before Easter and you're here and not 2 floors down over that side at the pub. So find out who is doing that or who is doing something close to that. Because a lot of the time, lecturers have a stack of ideas this big and they just can't find people to fill them, right? And there are so many opportunities out there that just aren't tapped into. So if you're interested in something, go and find a lecturer you think you get on with. Just have a chat with them. You never know, there's studentship money floating around. There's, there's all kinds of money, all kinds of funding, all kinds of projects floating around. And if it's not quite right, supervisors 90% of the time are willing to jiggle it a little bit. So in biomedical science, we do a bit less jiggling because what we do is really, really expensive and we have to find a lot of money. But often you'll find in other areas, there are supervisors who, do something to do with, I know Greg Howell, his students start off just with a species in mind. And then they basically watch the species until it does something interesting and that becomes their PhD, right? So there's a lot of fungibility in there. And if you put your hand up and say, yes, I am excited, and one of those comes up, you get an email, you agree to do it. And these opportunities are really great. So when I started off in this area, I interviewed with someone who used to work at SBS called Nigel Birch. And I sat down with him and we had an extended meeting that lasted for 20 minutes. And I understood absolutely nothing of what he said. I was applying for some studentship at the time. And I left and I wrote in the group chat, oh my gosh, I hope I don't get that job. I didn't understand a thing the guy said, I cannot manage this. About a month later, I got an email, congratulations, we're going to pay you to come and work for us for 10 weeks and do this thing you have no idea what it is. Which was really good. Turns out I really got on with him. It was a really good match. It worked out. I did a second summer studentship a year later on bacteria. Turns out I don't really like growing bacteria. Mammalian cells are much more interesting. So you kind of just have to guess and see who's around and who's got an interesting project and just do it for a bit. And if it doesn't work out, if it does, that's great. You end up doing a PhD. So apply. There is a lot of money hanging around. There are a lot of supervisors. They will talk to you, right? I know they look big and scary, but they will talk to you. And if you ever want to get a supervisor talking, just ask them what their research is. And it will go on forever. And you will very quickly know whether or not you find it exciting, because you'll either zone out in the first 2 minutes. Oh, yeah, I'm going to do that thing. So this is what it looks like. The reason I segue onto this is because this is what it looks like when you approach a supervisor. So this is the thing that you think all the supervisors know, and this is you, this little blue spot. You will find, once you get a little bit into postgrad, that in fact, it looks a lot more like this. You guys know more than you think you do, and you can find out anything. So often what you'll find is that there's a lot of people around you who know a little bit, but you know different things. So, do not be worried about lecturers. They're actually really nice. Go chat to them. Sometimes they'll buy you free coffee, and that's always good. So the next step is, how do you find somebody to work with? So you've come up with something you're really excited in. How do you know if you should go and work with that supervisor? And a lot of people kind of ask, who is a good supervisor? And it would be great if I had a list of good supervisors to give you. The answer is, there is no list of good supervisors. There is a list of supervisors, and there is a list of how they work. And it is all about the match. So if you go to, you guys won't be, but every year SBS holds this big Christmas party. And it's really interesting because the people who work together get on. So whenever we're hiring students, the first question that always gets asked after we meet them is, do you think you could tolerate sitting next to that person for a year, right? Because that's what you guys have to do, and that's also what the supervisor has to do. So you need to find a match. So this is a model that they hand out at the PhD induction, I quite like. So there are some supervisors who are really high structure, right? They want to know what you are doing every minute of every day. And some people find that very comforting, and some people find that they just want to be stepped through it. Some people find that is the most annoying thing ever, and they just want their supervisor to leave them alone, and they will come back to finish thesis in a year and a half, right? Those are these people over here. You also have supervisors who are very high support, right? These are people who want to know, are you sleeping well? Are you happy? Are you going home for Easter? Are you resting, right? That kind of thing. And you have quite a friendly relationship with them. And then you have some supervisors that are very low support. You get an email from them once a month saying, are you still alive? And when are you sending me the next paper, right? And it's all about the match, right? So what I would suggest you do, sit down with something like this and just think, what kind of supervisor would you like? Then you can ask the supervisors, what is your supervisory style? And they will just tell you. If you're too afraid to do that, go on the university directory, find one of their PhD students, ask them, and they will give you a very honest opinion. So yeah, just try and find a match for you. So if you can find an exciting project that you're excited about and a match, everything kind of starts to fall into place. And often you'll find that the things you find exciting and the supervisors you can get on with tend to overlap, because different scientific disciplines have surprisingly different personalities associated with them, without being too stereotypical. But it is interesting how people who do certain areas of science think in very different ways. And finally, a note of advice that I think you've heard from all the speakers, keep yourself broad, right? So jumping into postgrad, it's very easy to kind of just get absorbed by it. And that is a very unhealthy thing to do, because it never looks like this, right? It's never a case of gradually you move towards your goal, and it becomes positive. In fact, most thesis and postgrad journeys look a bit like this, right? They go up and they go down. And if all you have is your work and then your cells die, you get really sad. It is unhealthy how attached I get to my cells sometimes. We bought a new machine. That means I can look at them from home, which is so exciting. I won't do it now, but I can log on now and I can see them. And it is amazing how depressed you get when they all just slowly die over about a 6-hour period while you're supposed to be relaxed. Anyway, I will get off that topic. But the point is, if you have other things outside, it tempers that a little bit. So I've been quite involved with orientation and graduation and Toastmasters and a whole lot of things outside, right? Just because you're becoming a postgrad, don't drop that, right? And preferably find things that have non-scientists, because yes, T cells are exciting, but sometimes you really do not want to go over, like the guy who I was talking about earlier with the strong views on inflammatory T cells. Great for a Friday afternoon. Not so great when all your cells have died and you're already sad and you just want to whine. So balance is really important. The other really important thing is academically, as well as personally, keep yourself as broad as you can, right? So the students who we see who do really well are the ones who come in and go, oh, what are you doing? I think I'll learn how to do that as well, right? Because as a postgrad student, you're not just like a pipetting slave, right? It's not like an undergrad lab where they give you the protocol and go, do this 100 times, right? It's very exploratory, it's very open, and you're kind of just chucked in there with all the scientists to think for yourself. So if you have really broad interests, for instance, I was running an essay the other day, staining cells, and one of the master's students followed me doing that. And then a couple of weeks later, it turns out they need to stain cells and they already know how to do it, right? So if you kind of keep your interest broad, not only do you have more options going forward, but you also can kind of start to drive the project various places towards things you're interested in. So here are some of my side projects, none of which are very related to my PhD at all, but anyway. This is actually a project that I'm doing with one of the other PhDs. This is actually a recording of the inside of a rat, because you see this often. I saw it. I was walking through the building the other day, right? And I saw an eel in a bucket. Amazing. Anyway, I spent way too much time at this university. But anyway, this is the recording of the inside of a rat. And in blue are lymphatic vessels, which are what basically stop you from swelling up and turning into a massive elephant. And if they stop working, you swell up into an elephant. So his whole PhD is about how do we keep them working. So he's actually filmed the inside of this rat. And if I play it, you can see those little blue vessels occasionally kind of squish a little bit. It's very subtle, as you can see. So this guy has made 5,500 of these videos. And so I'm helping with some of the computing to count the little wiggles, which is proving challenging, but it's interesting. And I've learned a hell of a lot of things about high performance computing, but otherwise. The other one I've got up there, those are actually tuning blood preps. So if you want to get T cells out of blood, that's the stuff you use to do it. So I've actually managed to pick up a part-time job doing some of that for Rob Dunbar. So I work on a couple of clinical trials for various things. Again, if I hadn't followed someone doing a blood prep, I wouldn't have been offered that job. And the final one in the corner there is actually related to my PhD, but I took it this morning, so I'm still quite pleased with myself. So I thought I'd show it to you. So these are the cells that line inside of your brain. And the little blue dots, that's GAPI, which is actually DNA stain. So those are all the nucleus of the cells, you know, for biologists. And these lines here are all from CD144, which is actually a cell adhesion molecule that holds the cells to each other. So I took that photo, it worked beautifully. I was very pleased. And then the next photo in that series, I actually put on some steroid proteins and they all died. And it was great because they were supposed to die. It's always funny when you walk through the lab and someone goes, oh, the cells have died. And you kind of have to ask them, were they supposed to do that? Because half the people are trying to keep cells alive. The other half are cancer biologists. So they're trying to kill them. So you have to be very careful when someone goes, oh, my cells died. You don't want to go, oh gosh, I'm so sorry. They're like, oh, it's Anyway, I ramble. But yeah, have side projects, keep broad, and give yourself options, because, you know, over in the biology building, or wherever you end up, there'll be so many people doing so many exciting things. If you just be a bit nosy, you'll get dragged into all kinds of things. So, with that summary, good luck. You may need it. And yeah, just keep going, because if you really enjoy what you do and you follow what you enjoy, whether that is into a postgrad degree, and you should go into a postgrad degree, really good for the university's financial situation, and therefore I get more money to spend. Seriously though, whether it's, if you really have a drive in something you're interested in and you can find someone to share that passion with, go postgrad, you will really enjoy it. The same goes if you end up in industry, and all the guys have been talking industry and back again, right? No idea how your lives are going to turn out. I wish I knew. I could be really helpful, but I don't, and you don't either. So yeah, just keep pushing forward, do something exciting, and yeah, if you have any questions, I'm sure we'll do questions. Otherwise, I have a bazillion business cards that I printed for a conference, got cancelled because of COVID. So if anyone would like a business card, I will gladly give you one. Thank you very much.
Can we see your cells?
Can you see? Do you actually want to see the cells?
I really do.
Okay, here we go. It's only a graph though. It's not very exciting. Actually, yes, I think they're on. The problem with this machine is that if you leave it, it just keeps measuring. And so sometimes I left it over Christmas last year, and these cells very carefully hung on for about a week and a half, and then they all just died. And I came in the new year and just had this incubator full of dead cells.
Do you have to feed the cells anything?
Oh, you do. They get a media called EDM2MV, which is a mixture of 28 different components, and it's imported from the States and costs $1.50 a milliliter. So it's more expensive than whiskey. Yeah, it's a fortune, and technically it contains compounds that are not allowed in New Zealand, so I have to get a special permit from the government every time I use it. Yay! GMOs are a real pain. You can avoid working with GMOs, do. Good life advice. Any other questions?
Do the cells get names?
I'm not allowed to name them. They're called HCME-CD3, so they're actually a cell line. And so what happened was in the 1970s, there was a lady who was 28 years old, and she was diagnosed with a very severe form of epilepsy. And the treatment at the time was having half her brain removed. So they removed half her brain, and then they put it into a dish, and they spread it out, and they separated out those endothelial cells, the blood vessels. The problem with your cells, though, is if I, just take a bit of you and I put them in a dish, they die. So to stop them from doing that, they're actually been infected with a virus, and that virus makes them immortal, and they've been grown ever since. So yeah, they're all progeny of this woman's brain from the 1960s. And we freeze them under liquid nitrogen, we ship them around the world. We do all kinds of things. So no, they don't have names. It was funny a couple of years ago. So we've got this process whereby if you would like to donate blood, we take blood donors. It's all anonymous, but we kind of know who it is because someone disappears and then comes back with a box full of blood. And I'm like, definitely wasn't me. But anyway, so we did one of the master's students last year, and there is this particular subset of cells that are called naive T cells because they've never seen their antigen and been through an expansion process. And this guy had a massive number of these cells, like 5 or 6 times what we normally expect. So the joke about the next 6 months was, oh, you're so naive. Like, we can see it on this graph. It was a great joke. Once you get in a lab group, you kind of get on the in joke. So these are my cells. It's a god, no! All that suspense. Is it that one or is it this one? Please don't break it. The paperwork is awful. So someone got stuck in a door at orientation once, and it was 6 pages of documentation. So you want a fun read, there is a document that has all the health and safety accidents that happen in the university in a year, and they're all itemized by body part. 8 people had a lower thigh related injury last. So is that working? Or there are more remotes here. Isn't there more remotes than we have projectors?
This is a Sony projector and a Sony remote.
I turned it on.
It went right back off.
Okay, while the postgrads try and pick the projector, any other questions on anything specific, general, life in general?
Did you go to like
yes,
honours or PG Diff?
So the way it worked out for me was, so I did my undergrad, I got some studentship with Nigel. Then I did my 3rd year. I went to work for a guy called Reese Langley up the road on particular bacteria. Lovely guys, but I just couldn't get excited about Staph aureus, just me. So then I did my honours with Nigel on that T-cell project. And then basically towards the end of that, we had some really interesting findings on how blood proteins interact with endothelial cells. So basically I wrote a list of things I wanted to do. It became a grant. I got offered some money, and that's been paying me for the last 3 years. So there's a lot of latitude to do stuff like that. So if you reach out to people, there's a lot of these grants that turn over. And often it's, for instance, one of the guys I was talking to the other day really wanted to work with CRISPR, the editing technology, and he really wanted to do CRISPR on T cells. There wasn't any money for that. So he's doing CRISPR on skin cells. Same thing, right? But yeah, there's a lot of stuff out there. Oops, not asleep. So this is we've got a big tissue culture suite, which basically is a room about half the size full of massive hoods that are sterile because these cells have no immune system. So if you breathe on them, they will die. And these are all my cells. So there's half a plate's worth, so 48 of them. And then they've come up, and then I've given them various treatments. And you can see these ones are unhappy and have died. These ones got whole human blood, and they're very happy. So they've grown up, and this has been running 167 hours. And it's great because this is actually a live view of the screen in the tissue culture room, which is great because you can go and write people messages in the middle of the night. Sometimes they write me messages. I came in one morning and they said, "Your thing is beeping, please." I do have a mobile phone. They could have called me, but no, you won't be looking at this. It's kind of sad. Any other?
How do you go by like applying for the summer studentship stuff?
How do you go about applying for summer studentship? They all go up, and so what happens is roundabout. It depends because COVID has stuck everything. But round about the start of first half of semester 2, there is an email goes around the school and says, does anyone want a summer student? And from that, a big list is compiled of potential projects, and that is then put on the university website. So if you look up there, if you just Google University of Auckland Summer Studentships, there will be a mess. You can look at last year's. All kinds of things with all different people. Basically, you email the supervisor and say, hey, I'm interested in your project. Then you put in an application, you put in your CV, you write a little blurb about yourself. Then there's a massive and very complicated funding process that takes months because there are a whole lot of different people who want to fund different things and different pools of money and total mess. But anyway, from your end, supervisors, sometimes they reach out and say, we'd like to meet you. Sometimes you just get an email, can be any of those things. And then, yeah, you basically get offered a scholarship like any others. It goes on your transcript. I think it was 6,000 last year, 6,500. So it's minimum wage, but it's minimum wage after tax, pretty much. So basically, you're paid to be there for 10 weeks to work on a project. A lot of them are kind of pilot projects. So they're things that supervisors think are interesting, but I'm quite sure if they're going to work. But yeah, highly recommend. Makes a really good summer job, and it's a really nice way to have a look at postgrad and get paid without committing. So they are quite competitive, but by the same token, there are quite a few of them. that go. So yeah, reach out to supervisors. Sometimes they can just, a lot of the time, the way the university works is that at the end of the year, if you have money and you haven't spent it, you lose it. So towards the end of the year, there's a lot of supervisors who have a couple of grand left off the end of various operating accounts. And sometimes they can just go, yep, fine, come work for me. So yeah, really, really exciting thing. So worth applying for.
Yeah, and don't be afraid to apply for ones outside of your department or faculty as well. I've had friends in doing Bachelor of Medicine and Surgery, and they've actually done ones through optometry and got to work with optometrists for a summer. And same with students who are doing biological science, they've done chemistry or marine science. So don't be afraid to apply for ones that you haven't done undergrad work for. Yeah, it's, if you're passionate and you're keen to try something new, you've got a good shot. So, yeah.
I'd like to add to that as well. So a lot of these summer students of summer scholarships have the position supervisors have like a listing of what the project may be, and then a bit about what kind of they're looking for. If you don't have that skill set, don't let that deter you. Apply for it regardless, because trust me, they will be looking for someone. Just because it says we need someone who's proficient with R coding doesn't mean they need someone with R coding.
Mm-hmm. And so I had a summer student over last summer, and to be honest, the reason we picked the candidate we did, it's not here, so that's all right, was because we pick students. So when I say that, what I mean is we pick people who we think are interested in the project, people who we think we can get along with, and people who share our values, to use a really open term. Right? So we pick students, we don't necessarily pick skill sets. Some lecturers say, this project is all about R stats, and I teach the paper on R stats and it's really good, right? Some lecturers cannot be taught about that because they taught their paper and they want someone if they taught their paper. Other lecturers are just like, you sound interesting. But again, if you have interests in things outside, it all looks really good, just holistically, because at the end of the day, even lecturers know that the list of lectures on your transcript is not even close to a good representation of you as a student. And I know several of the most successful PhD students who I work with, brilliant scientists, useful undergrad grades. So just because you having high grades will make it easier to get a position, because that's how the money is handed out, unfortunately, because it's the fairest way to do it. But just because you're not an A+ student, absolutely does not mean you shouldn't reach out and apply.
There is a GPA of 6 minimum for non-Māori and Pacifica, and then Māori and Pacifica is 5, I believe. 5.5.
For summer studentships,
yeah, I think
most of the
minimum.
time.
Yeah.
So there are a whole lot of pots of money. The best thing to do is reach out. If you're interested, reach out to the supervisor. Sometimes they'll say, yeah, apply for a central scholarship, in which case a 6 is going to really help you. If they can't do that, there are other ways, there are other things. There are project stuff around the lab. If you're doing science scholars or project proposals or anything like that, there are lots of ways that you can work with people. So flag your interest, and then when the opportunity comes up, let me know. But yeah, there's a whole lot of GPA kind of stuff. But again, if you flick the lecturer, I am interested in your GPA, they'll know all that off the top of their head and immediately be able to go, yep, or no, not quite right for me, or there's a lot of shuffling that happens behind the scenes because we all sit next to each other and it's like, I'm a really good student, but I don't have space.
And a motivator to do masters, if you're doing a research masters and you've done honours or PG DipSci, if you get a GPA of 8, you get a very nice $24,000 scholarship automatically that you don't even have to apply for, which is very nice. So you get $14,000 to help your rent plus free course fees, which is $10,000 off your student loan, which is really nice for those of you thinking of going on to masters. And there's similar ones for PhD, which is it 24,000 a year?
Honours do have scholarships, but the GPAs are quite high. PhDs, you can either get an automatic scholarship, which I think pays $28,600 this year, tax-free. So that's equivalent to just under $40,000. That also pays your fees. However, if you don't hit that, there's also a lot of external money. So for instance, Indy have a whole load of stuff. And if you basically reach out to supervisors, some of them will go, oh yeah, I've got, sometimes Indy hand out, how big's an Indy grant nowadays? It's like $1.3 million. Like that's on the low end. Marsden's are anywhere up to $2.4 million. So paying you a stipend to come be their student for a year, some of these projects is not an issue. So reach out, they will know, and there are emails that go around the faculty. So if they don't know, they will know someone. Money. No more comments, questions, complaints? No. Excellent.
Sweet. Thank you for that.
That's all right.
Yeah, so no more questions or anything? Yeah, I'd like to say a huge thank you to all the speakers that took the time to come up and talk tonight. It was really good. And I'd like to say a big thank you to Decisive for also advertising the event. And yeah, we've got cold drinks, which are in the fridge, which I'll bring out. And we've got pizza and some hummus and stuff. So if you guys would like to stick around and network for a bit, we have some of that. But yeah, otherwise, thank you very much, and hope you have a good evening.
Yeah. Oh, sorry, I can like crop this out.